Hello viewers, I am Dr. Om Salakari, working as an Assistant Professor in Department of Pharmaceutical Sciences and Drug Research, Punjabi University, Patiala. Today, I am going to discuss about the traditional methods of drug design. Basically, process of drug design is a complex process because of the complexity of physiology of human body. It is a time-consuming and labor-intensive process. This lecture designed in 6 modules. Module 1, Introduction. Module 2, Isolation of bioactive compounds from natural sources. Module 3, Accidental discovery. Module 4, Random screening. Module 5, Modification through disjunction and conjunction. And Module 6, Crosstalk between traditional and modern methods of drug design. Now, I start Module 1, Introduction. Drug designing is an integrated developing disciplines which pertain an era of tailored drugs. It involves the study of effects of biologically active compounds on the basis of molecular interactions in terms of molecular structures or its physicochemical properties involved. It studies the process by which drugs produce their effects, how they react with the protoplasm to elicit a particular pharmacological effects or response, how they are modified or detoxified or metabolized or eliminated by the organism. Traditionally, drug molecules are discovered by the number of ways. Although traditional methods have been replaced by various new techniques like combinatorial chemistry, high throughput technique, computer aided drug designing techniques, etc., these are still play indispensable role in modern drug discovery and development processes. Module 2, Isolation of bioactive compound from natural sources. Basically, sources of drug molecules are from natural origin, especially from plant. On the basis of observed pharmacological activity, one can isolate active constituents as drug and can also optimize active constituents to obtain semi-synthetic drugs. The first example is decomoral, which is a anticoagulant. In 1921, it was shown that improperly dried clove hail gave rise to internal bleeding in cattle. In 1934, the compound responsible for causing the internal bleeding was identified as decomoral. In 1941, decomoral was introduced into medicine as an anticoagulant. Decomoral competes with vitamin K, and since vitamin K is regarded for the synthesis of prothrombins, the clotting of blood is produced, and therefore this compound finds use in such disease as thrombosis. The structure activity relationship of the decomorals reveals that for anticoagulant activity, the following structural requirements were necessary. The first one is 4 OH coumarin rings, and second, at position 3, a hydrogen or alkyl substituted alkyl group was required. From this lead molecule, improved anticoagulants were discovered, that is warfarin. Other synthetic drugs which were discovered based on bioactive natural products include the following: synthetic QSAR-I drugs based on tuberculin, local anesthetics from cocaine, antimalarial from quinine, antimicrobial from penicillin and tetracycline, and anticholinesterase from phytostigmine. Module 3, Serendipity or accidental discovery. Serendipity in drug discovery implies the finding of one thing while looking for something else. The Oxford English Dictionary defined it as the faculty of making happy and unexpected discoveries by accident. In Stedman's Medical Dictionary, serendipity refers to an accidental discovery, for example, finding one thing while looking for something else. Serendipity refers to chance discoveries that have been exploited with the sagacity. This requires both a chance event and the mental ability to understand the occurrence and realize its potential. Serendipity is one of the many factors that may contribute to drug discovery. To calculate the proportion of drug with serendipitous background, the total number of small molecule drugs on the market FDA approved is taken to be 1437 according to Drug Bank. The serendipitous events were divided into 2 categories: laboratory based and clinical observation based. First example is the serendipitous discovery of penicillin in 1928 by Alexander Fleming. Fleming was engaged in research on influenza when one of his Staphylococcus culture plates had become contaminated and developed a mold that created a bacteria-free circle. Fleming recognized the possible significance of the bacteria-free circle, and by isolation the mold in pure culture, he found that it produced a substance that has powerful destructive effects on many of the common bacteria that infect men. Penicillin. When acetonolide was incorrectly dispensed from the naphthalene, the antipyretic property of acetonolide were noted. From the lead molecule, phenastine and acetaminophen were discovered. Another example is serendipitous identification of sulfanilamide. Basically, Paul Ehrlich had the idea of preparing dyestuffs with selective affinity and toxicity for bacteria. From this investigation, Prontosil was discovered as a very useful antibacterial agent. However, its activity was due to its metabolites sulfanilamide. Another example is Librium, the first benzodiazepine based lead as tranquilizer. A series of quinazoline 3-oxide was synthesized by Dr. Leo Steinbach at Roche in a program to develop a new class of tranquilizer drugs. Since none of these compounds was found to be active, the scheme was terminated in 1955. However, a vial from the above scheme, which remained untested, was found in 1957 during general laboratory cleanup. The compound was submitted for pharmacological testing to complete official formalities. Surprisingly, it gave a very promising results during preliminary screening for tranquilizing activity. It was found to be benzodiazepine 4-oxides, presumably produced in unexpected reactions of corresponding chloromethyl quinazoline 3-oxide with methylamine. Obviously, chance played a major role, but the intelligence and skill of the scientists who were prompt to catch the fortune should not be disregarded. In any case, even it is always welcome, this strategy does not really seems to be one to rely upon modern drug discovery. Module 4 is random screening. Random screening is most popular method of traditional drug design. Basically, random screening means the bioevaluation of compounds. This can involve examination in one or many test systems and is justified in following two cases. First case, in the search for new classes of compounds which have the desired biological activity. Often, this is tried when molecular modification of existing drugs is unable to improve the activity of drugs. Second situation is where the biochemical reason for the disease is unknown. Random screening is justified because one cannot design a compound to interact with key receptors. Screening of large number of synthetic chemical compounds or natural products for desired effect is traditional way to design new drugs. It is not ideal one for number of reasons. First one, it requires an appropriate screening procedure. Second, it is usually inappropriate to put the chemical of unknown efficacy directly into animals. Third, animal model may not accurately reflect the human diseases. Fourth, the chemical may be extensively metabolized to different compounds in animal before reaching its target. Fifth, the chemical may not be absorbed or distributed as it is in human. And finally, because of its random nature, it is inherent, repetitious and time consuming just to find a chemical of desired activity. Major advantages of screenings are the large amount of information that is not needed to carry out the process. Second, one does not need to know the structure of drug being sought. Third, one does not need to know the structure of target upon which the drug will act. And finally, one does not need to know the underlying mechanism of the disease process itself. Module 5, Modification through disjunction and conjunction. Once the active compound has been identified and its chemical structure determined, it is usually possible to improve on the activity and or reduce its side effect by making modification to the basic chemical structure. Modification to improve the performances are often carried out using chemical or biofermentative means to make the changes in lead structures or its intermediates. Alternatively, for some natural product, the gene itself may be engineered so that producer organism synthesizes the modified compound directly. The cordial objectives of the method of modification are: first, to improve the potency; second, to modify specificity of action, to improve duration of action, to reduce toxicity, to effect ease of application or administration or handling, to improve stability, to reduce cost of production. The example of this approach is in anti-infective area, where modification of the original first generation cephalosporin have led to second generation and third generation offspring with substantially improved characteristics. It can be done in two ways: first, modification through conjunction; second, modification through disjunction. Now, first, modification through conjunction. This is known as systematic formulation of analogues of a prototype agents in general towards structurally more complex products. In this type of drug design, the main principle involved is the principle of mixed moieties. A drug molecule is essentially made up with two or more pharmacophoric moieties embedded into a single molecules. For example, ganglionic blocking agents, its development based on the principle of mixed moieties. The principle of mixed moieties actually involve the conjunction of two or more different type of pharmacophoric moieties within a single molecules. Now, second one is the modification through disjunction. Modification through disjunction is a systematic formulation of analogues of prototype agents in general towards structurally simpler products, which may be viewed as partial or quasi-replicas of the prototype agent. This process aims at providing the compounds with retention of activity but removing the unwanted side effects. The method of disjunction is usually employed in three different manners, namely unjoining of the certain bonds, substitutions of aromatic cyclic system for saturated bonds, and finally diminution of size of hydrocarbon portion of the parent molecules. For example, extensive study on the estrogenic activity of estradiol by drug design through disjunction ultimately rewarded in the crowning success of synthesis and evaluation of trans-diethyl stilbestol. Now, merits of this method: one does not need to know the structure of target on which it will act. Also, no information regarding underlying mechanism of the disease is required. Second one, as with the screening, the process of modification is often based on trial and error approaches because more information is known. However, this process can be carried out with much greater probability of success than purely random process. Now, the demerits. The whole process is based on the single lead compounds for further drug designing. If the original lead compound fails to generate a desirable drug, one must start the process over again by finding a new lead molecules. Improvement are likely, however, no major breakthrough in developing new chemical entities is probable. Now, in module 6, crosstalk between traditional and modern methods of drug designing. Nowadays, advanced mechanism based computer aided drug designing technique is currently being used to design new molecule as drug. When mechanism of disease process is understood at molecular level and target molecules are defined, one can design a new molecule as a drug or optimize the lead molecule for desired pharmacological activity or designed pharmacokinetic profile using either receptor based or ligand based drug designing. Now, first, receptor based drug designing. When 3D structures of the receptor is determined either by X-ray crystallography or NMR or homology modeling, one can design new molecule as drug by directly anticipating the requirement of complementary ligand by analyzing interior architectures of the active site in it. For example, docking analysis is the most commonly used receptor based drug designing technique of structure based drug designing protocol. Second, ligand based drug designing. When 3D structures of receptor is unknown, one can design new molecule as drug by comparative analysis of already existing active, moderately active and inactive molecules along with their biological activity. For this purpose, physicochemical or structural properties are correlated with biological activity to generate hypothetical receptor model, which can be further utilized in designing of novel molecules or can also be used to optimize pharmacodynamics and pharmacokinetic profiles of drug molecules. For example, traditional QSAR methods including Hansch analysis, free Wilson analysis, second 3D QSAR analysis like comparative molecular field analysis, comparative molecular similarity index analysis, receptor surface analysis, molecular field analysis, and third one is semi-quantitative methods like pharmacophore mapping. With the introduction of these CAT technique and integration and knowledge management solution, a new era of beginning in drug discovery. The development time are reduced from 10 to 15 years to only 6 to 8 years. The new approaches to drug discovery exceeds the limitation of traditional methods, but we cannot rule out traditional methods completely. Traditional methods and modern techniques are used in combination to overcome the demerits of both methods. Basically, modern techniques serve as a tool for traditional methods and therefore accelerate these time-consuming process. Now, this chapter of traditional methods of drug design can be summarized as: drug designing is an integrated developing discipline which pretends an era of tailored drug. Traditionally, drugs have been designed using following methods. First, isolation of bioactive compounds from natural sources. Second, accidental discovery or serendipitous way of drug design. Third, random screening. Fourth, modification through disjunction and conjunction. Number of drugs have been developed using these methods that are now used clinically. Despite of successful examples of these methods, they have many demerits associated with them. Traditional methods are extremely time consuming and cost intensive. To overcome the drawbacks of traditional methods, new advanced mechanism based approaches have been introduced. The development times are now reduced from 10 to 16 years to only 6 to 8 years. Thus, traditional and modern methods can be integrated to design new drugs in rational and cost-effective manners. This chapter highlights detail about the important traditional methods of drug design along with its role in modern drug design techniques. Thank you.